Have you ever stopped to think about what's happening inside your own cells right this second? I mean, it's not just some empty bag of goo, it's a universe in miniature, just packed with these tiny, incredible machines all working together. Today, we're going to take a journey right into that universe. You know, a good way to think about a single cell is to picture a bustling city. You've got the city hall, which is the nucleus, you've got power plants, the mitochondria, factories, recycling centers, the whole shebang. So what if you're a biologist and you want to understand just the power plants? You can't really get a good look with all the noise and chaos of the rest of the city getting in the way, right? So the big question is, how do you isolate just one part of this tiny microscopic metropolis?
You see, to figure out what goes wrong in diseases, or really just to grasp the fundamentals of how life works, we have to be able to study these little city components, the organelles, all by themselves. And that's where this amazing technique called cell fractionation comes in. It's this brilliant way of, well, deconstructing the city so we can actually read its blueprints.
Okay, let's really get into this. At its heart, cell fractionation is exactly what it sounds like. You're breaking a cell into its fractions, or its parts. It's a super important technique in biology that lets scientists take, say, a piece of tissue and end up with pure test tubes of just nuclei or just mitochondria. It's all about taking things apart to see how they tick.
So how does this all start? Well, the first step is actually pretty straightforward, at least in concept. Before you can sort anything, you got to get it out of the cells. You have to make what I like to call a cell smoothie.
The official name for this is homogenization. What happens is scientists take the cells and pop them into this special liquid. It's kept really cold, and that's important because it stops all the cell's own enzymes from basically chewing up the very parts we want to study. Then they use something like a high-tech blender or even sound waves to break the cell membranes apart. This lets all the guts of the cells spill out into the solution, creating this thick soup called the homogenate.
But here's the key. This isn't about just brute force. It's more like a controlled demolition. You have to be gentle enough to crack open the outside of the cell without totally wrecking all the delicate and precious machinery inside. The goal is to set them free, not to smash them to bits.
All right, we've successfully made our cell smoothie. But right now, it's just a jumbled mess of all the cell's parts floating around together in a tube. So the next and maybe the most critical step is to sort through that mess. It's time for the great separation.
And the tool for this job is the centrifuge. Now, if you've ever been on one of those spinning rides at an amusement park, you know that feeling of being pushed to the outside, right? Well, a centrifuge does pretty much the same thing, but for tiny particles and at mind-boggling speeds. It spins these tubes so fast that it forces everything inside to separate out based on how big and heavy it is.
So here's how it works in practice. You take your homogenate, put it in a tube, and spin it. The heaviest, densest things, like the cell nuclei, get thrown to the bottom first. They pack down into this little solid lump called a pellet. The rest of the liquid, which we now call the supernatant, still has all the lighter stuff floating around. So you can carefully pour that liquid off into a new tube and get ready for the next round.
Now that basic idea, spin and separate, can be done in a couple of main ways, and it really just depends on how picky you need to be. So let's take a look at the two major techniques that scientists use to sort the contents of our cell smoothie.
So on one hand, you have differential centrifugation. This one is all about speed. You just keep spinning the mixture faster and faster in different stages, and with each increase in speed, you collect smaller and smaller organelles in your pellet. On the other hand, there's density gradient centrifugation, which is, well, it's a bit more elegant. For this one, the organelles are spun through a special liquid that's layered, kind of like a fancy drink, getting denser toward the bottom. The little organelles travel down until they hit a layer that perfectly matches their own density, and they just stop, forming these perfect, neat little bands.
So check this out. This table really shows how it works. You start with a pretty gentle spin, only about 600 times the force of gravity. That's enough to pull out the absolute heaviest things, the nuclei, into your first pellet. Then you take the leftover liquid and you crank up the speed, a lot, to 15,000 Gs. Boom! This time you've isolated the mitochondria, and you just keep doing that, spinning faster and faster to get smaller and smaller things, like microsomes at an insane 100,000 Gs.
So after that whole series of super calibrated spins, a scientist can actually hold up a test tube that has a pure sample of just one type of organelle. They've done it. They've successfully pulled just the power plants out of the cellular city, and now they can study them up close with no distraction.
Look, this is a really cool and clever procedure, for sure. But it is so much more than just a neat lab trick. This technique has been absolutely essential to, well, almost all of modern biology and medicine.
So let's talk about why this all matters so much.
I mean, think about it. By pulling out just the mitochondria, we can see how they fail in diseases like Alzheimer's. By isolating ribosomes, the cell's little protein factories, we can design and test drugs that stop bacteria from making their proteins. We can figure out exactly which part of a cell is being messed up by a genetic disorder. This technique literally turns the cell from a mysterious black box into a puzzle we can actually solve.
And really, it all boils down to this fundamental philosophy in science. A living cell is just staggeringly complex. To even have a chance at understanding how the whole system works together, you have to first understand the individual parts. And cell fractionation is the tool that gives us the power to do exactly that.
So, from the most basic biology to the most cutting-edge medicine, this journey inside the cell has shown us so much about what makes us, us. It's a great reminder that sometimes, to understand the biggest picture, you have to start by looking at the very smallest parts. The secrets are in there. They're just waiting for us to sort them out. Which leaves us with one question, right? What are we going to find next?